Good morning.
Good morning, I'm Asim.
Hi, I'm Neeraja. Welcome to the Sunday series at Science Activity Center, IISER, Pune. As you know, the Indian Institute of Science, Education and Research is a premier institute to investigate or explore various basic sciences. So we have programs in physics, chemistry, mathematics, biology. We have programs in data science, earth science, and also in science education now. And all of you can come here and, explore science with us. Today, we are here to celebrate the Children's Day, which is really tomorrow. But since this is our Sunday series, let's try to explore the puzzle of 6CO2 and 6H2O and how does it really come to give us the sugar and oxygen.
But before that, let me give you a virtual tour of IISER Pune. So this is our green campus. Lot of trees, lot of science and lot of activities going on here. You can see the overview in this particular picture. Along with virtual tour, of course, you can visit IISER Pune. You can see here, this is our main entrance, and you can also see our motto, where tomorrow's science begins today. And this is our own Science Activity Center, where we are sitting inside. So you can come and visit this. What will you do when you are here? Of course, there are lots of activities and science camps happening. You can be part of this. You can learn science in many different ways. Not just you can also see these pictures where you can see your teachers also coming here. So this episode, I know our teachers are also watching. So you can also come here and learn and develop and design different activities that you can use in your own school and your own classroom. So coming back to the puzzle that we are talking about, Neeraja did mention what the episode is about, basically solving the puzzle. But Neeraja, don't you think this is already there in your textbook?
I think it's already there in the textbook,
What else
right?
is there?
I think there are many other things in your textbook. So if you open your science textbooks and whenever you say I want to study science, we let's look at all the things that you study. You study about Newton's laws, you study about gravitation, laws of motion,
Physiology.
Right, you have various equations, chemistry equations. You study about combustion reaction. You study about photosynthesis, respiration. So many things, right? The textbook is full of these things. So when I say I study science, I should just open these books and start studying, right, Asim? Is that how?
Nothing else to do, right?
Okay, so this is what we want to talk to you about today. Is how do we really study science? And in order to show you how science is really studied and how science is really explored, we are going to talk about one case study, which is this particular reaction that we always see in our textbooks. 6CO2 plus 6H2O giving us C6H12O6 and oxygen. How does it really, how did we come to know this, right?
I can just imagine this, thinking about those people and scientists who have worked on this and they have developed basically answer to the puzzle.
Right.
But when did this happen? Do you know?
So to think about it, we have been trying to solve this puzzle for last 400 years and it's still not completely solved. And last 400 years, not just one scientist, but more than 40, or rather, I will be surely wrong, but 40 main scientists, I would say, have explored this puzzle and tried to figure out what is the process of photosynthesis.
I think that is the beauty of doing science.
Yeah, like it never ends. You keep on exploring, and that's the fun part of science.
So the timeline that we are thinking about, where actually the investigation to solve this puzzle started, is around what time?
So we are talking about early 1600s. That's probably the time when we started exploring the life around us, and somebody questioned how do plants grow.
That's brilliant question to ask, especially in 1600.
Yeah.
But when you say 1600 or 17th century, what else was happening there?
So let's think about early 1600s. So let's kind of just close our eyes and think about we are in early 1600s.
Okay.
So what is around you? So I think this is the first time the Jupiter's moon was seen by Galileo in a telescope. First time I think we saw something that is outside Earth using a telescope.
Wow.
That's the first time. Then how to calculate. So there was a lot of work going on in calculations, mathematics per se, how to add really large numbers together or how to multiply really large numbers together. So logarithms were first.
Yeah, I remember. So this calculus that we study in school and then in college, I
Right.
think started in that time.
Early 1600s it was
Right.
started.
But what was happening in India at that time?
So Indian science had we always say that Indian science has progressed, and we really knew a lot of things in geometry, we really knew a lot of things in mathematics.
Right.
If you think about the main events happening in India, I think Shivaji Maharaj really, getting crowned as a king.
Yes.
It was in 16th century, or Taj Mahal being built was
Wow.
that era. So we are talking about that era. So imagine you have no fancy tools for doing science. You don't know
Absolutely.
what a microscope looks like, or you
No.
don't know. Telescope was the first big instrument that really came to
Exactly.
be.
Exactly. So no mobile phones,
No
no
mobile
internet,
phones, no internet, no Google,
nothing.
nothing.
Electricity?
No, not
Perhaps
yet.
not.
Not yet.
So our scientists started solving this puzzle so long ago. So it will be interesting to see how they come up with their answers and what sort of experiments they did.
Right.
So can we take our audience, our students and teachers on a tour?
Okay, so let's imagine us to be a part of that era, and
Okay.
let's imagine that we are in 1600s trying to ask the question, how do plants grow? Because there's so many plants around
Yes.
us. It's a
Yes.
good question to ask. Before we answer that question, I want all of you to really tell us in the poll, where do you think most of the biomass of a mango tree, that like we see a small mango plant, and then it grows to a big tree, where do you think the biomass really comes from? Does it come from soil? Does it come from air, water? We have to water the plant, or does it come equally from all the above? So where do you think most of the biomass comes from?
And we wanted to think first. The fastest answer will not fetch points. The correct answer will.
Okay.
Just type in the chat.
Really good answers in the chat, right?
Right.
So most of them are, I think, trying to guess where does the main part, or where does the main biomass come from? Does it
Right.
is it soil, Asim?
I think so, because all plants grow on soil. I don't think there is anything in air. And
Okay.
water is just water, right?
Okay, so I mean that's probably the thing that the early 1600 scientists kind of tried to think, is that we know that soil is required, we know that water is required for plants to grow, so mainly these two things should contribute, right?
Neeraja, I can see also some answers which are coming, which are based on the photosynthesis that we know now.
Okay.
But I think we have to ask our audience to actually go back in the 17th century and imagine yourself being there, where you have no idea, no textbooks, perhaps no schools. So let us go back again there and think about what were scientists doing and what sort of answer they were actually giving to this particular question that Neeraja has asked.
So, when this question was first tried out, it was the most popular belief was all the good things are made by God.
Yes.
So, of course, plants growing is a good thing. We get food. So, God makes plants grow. That's the
Okay.
first thing you will say, right? I mean, everything bad is by the devil, everything good is by the God. So, God is good. It gives us, or he or she gives us food, right?
But some of us during that time also thought, yes, I understand God. But we are also sort of made by God. But we do
Okay.
eat, and that's how we grow.
Okay.
So some of the scientists and some of the people who were working on this question at that time also started thinking plants eat soil, and that's how they grow.
Right. So that's one way to think about it, just how we eat, plants eat soil. So now if we want to think about the puzzle, we have soil, water, making plants grow.
Grow, yes.
Okay. So the first person to really tackle this was the scientist named as Jan van Helmont. And he was actually a person who believed in medicine, who believed that if somebody gets ill, they get cured by something medicinal and not by God's grace.
Okay.
So he was actually questioned by the church a lot, saying that you're going against the God. So he was kind of kept under house arrest. So all the experiments that he could do were kind of the experiments that we'll be able to do sitting at home.
Yeah.
So you can also try this at home.
I think
What?
our students also know Galileo's story and other stories
Yeah,
where
so
they wanted to do science, but perhaps the environment was not really
not
conducive.
true.
Okay, but then what did Helmont do?
So Helmont thought that, okay, is it the soil that is making plants grow? Because he knew water was important. Without water, plants died.
Yeah.
But for soil, what he did was in his garden, being under house arrest, he planted a small willow tree. And before planting, what he did was he measured the soil.
Okay.
And not only just the soil, he dried the soil to make sure that there was no water weight.
So only soil he.
Only soil. And then he planted the tree, let the tree grow for, good 5 years. And then later on, he measured the weight of the tree.
Did you say 5 years?
Yeah, so the
He
experiment.
did that experiment for 5 years?
Yeah, so the
Wow.
experiment lasted for 5 years. In fact, for all plant experiments, I think we have to be patient because
That's true.
they work really slow.
Yeah.
So afterwards, now the tree is grown. He's measuring the weight of the tree. He also measured the weight of the soil.
Okay.
Again, he dried the soil to measure the weight to make sure that water is not really getting in the
I
way.
think that's a sign of very good scientist.
Yeah.
Everything is measured and very accurately.
Right. So what he observed, what do you think he would have observed?
I think he observed plant grow.
Okay. So
One option is the soil weight went down because
Okay.
like we thought at that time, people were thinking plants eat soil,
Mhm.
and so soil weight went down. Or soil weight remained as it is because perhaps the answer is not just soil which helps plant grow. Perhaps there are other things.
Okay, so that's actually what he observed. So the
Okay.
soil weight didn't change much. It was barely changed throughout
Okay.
the experiment, but the plant weight kind of went, more than 30-40 folds.
Wow.
So that's when we knew that it is not the soil.
Okay.
Okay, so then if you were Helmont, what would you say?
Perhaps water is
Oh.
the correct answer.
Okay, so that's what he thought. He thought that it is mainly the water, not the soil, that plays a role or gives the biomass to the plant.
Okay. Neeraja, I remember during that time, of course, many other scientists and investigators were interested in plants, growing
Right.
plants. This was a time where especially the European countries, especially England and Netherlands, etc., they were going out and trying to also get plants from other countries and try to grow in their own
Right.
countries. Now, for doing that, of course, botanical gardens were really famous.
Right.
So even though Helmont did all his experiments separately because he was afraid, but of course, a lot of interest was generated in growing n number of plants from different countries. But you can then imagine the weather in England compared to the weather in our country is completely different.
Yeah.
So people started building different enclosures
Okay.
where they could grow different plants, which could maintain, those enclosures could maintain different environmental conditions like heat, humidity, and so on. But this is still the time where you are saying we were still investigating what makes plant grow.
Right. So
I'm
we
going
had
to share with you all a very interesting map of Oxford Botanical Garden. So these are actually the plants, like the architects make. So you can see different gates at different locations. You can see, of course, a very beautiful garden plant. But here somewhere, you can also see a separate house, that is house for plants. Now my question is, do you think there is something wrong in this? Let us ask our audience. You can type in the chat.
Yeah, what is wrong in this particular building?
This is house where we live. right?
It's a house
This is
for
house
plants.
for plants. Anything wrong in this? Okay, some answers are coming in. Yes.
Okay, so some people have actually guessed it right? It's inside, it's closed.
Right. So what's wrong in being closed? You can give water, you can give soil.
So actually, it tells us that we had no clue that
Exactly.
sunlight plays a role in
Yeah.
making plants
Even
grow.
though this was Oxford Botanical Garden, the plants which were kept inside hardly grew.
Right, so this was a failed experiment. then.
Exactly. But still, failed experiment can give you lots of ideas for new experiments. Now, this is where we come in the mid 17th century. Neeraja, you want to talk about his story and his discovery?
Yeah, so Joseph Priestley. So this is something that even our audience knows because it's in the textbook. Like
Yes.
how everybody was kind of studying gases at that time.
Yes.
In fact, we didn't know that air is a mixture of gases until 1700s. We used to consider air as one entity.
Yes.
And oxygen was not really discovered, but we had some idea that there is something in the air that is essential for us living,
Okay.
and it is also essential for combustion.
Something good in the air.
Something good in the air. So in
Okay.
fact, the good air had oxygen in it,
Okay.
and the bad air was when the oxygen was removed. So all of us have done this experiment in class, wherein we have this candle. When we invert a glass jar on it, what happens? After a while, the candle is going to extinguish,
Yes.
and you're going to see some smoke. But what really, according to Priestley, happened was that we have removed all the good air, and now the bell jar is full of bad air.
Okay.
We still don't know that it forms carbon dioxide.
Yes,
Right?
because we are still talking about 17th century.
Right. No idea what really forms.
Yeah.
But it's good air and bad air that we are talking about. So what Priestley did was he also tried to go beyond this particular experiment.
Exactly.
And he said, we knew that the bad air is bad for mammals living. So they had actually tried mice putting in the bell jar. The mouse actually died, indicating that we can't live. But he thought, okay, what about plants? Can plants live inside that bell jar?
Yes.
And he put a mint tree inside, and it surprisingly, grew really nicely inside that bell jar. So what is the case now? So
I think our students now know the answer, but we are talking about 17th century. We are talking about a time where people did not really understand what oxygen is, what carbon dioxide is. But see, this observation is very useful. That he could at the end conclude there is something in the air, not the soil, but air that is useful for plants.
Yes, so plants actually are going to probably take the bad air. That's what he concluded, and that's actually interesting how he concluded that, using the next set of experiments wherein he put a living mouse and the mint tree together and the candle together in a big bell jar, and showed that even when the candle extinguishes, even when there is bad air, there is something given off by the mint tree, which is basically making good air now.
Right.
So and that's why the mouse lives and doesn't die.
Okay.
So that's really interesting.
I'm sure I mean, everybody is looking at a drawing here, but I'm sure he must have used a really huge jar where there is a lot of possibility of generating oxygen and a huge tree also. Of course, we are not going to recommend our students and learners to actually do this at home. But there are a good amount of videos available where this experiment is explained very nicely. This is very important because now Priestley is able to say, based on what has happened till now, that plant can cure air.
Right.
Because of the mint tree, the mouse could survive. So there is something plants are producing. Rather, let me take. There is something plants are taking from air. They are using it. They are producing something which is perhaps useful for us.
Probably true. And in fact, in that era, a lot of experiments were done on gases.
Exactly.
So not only this, but good air was also being produced by, let's say, burning mercuric oxide. And it also produces oxygen. So people thought, so there were multiple other things. In fact, people had started thinking that plants give off good air.
Okay.
So there is a gas that comes from
plants.
But how do we observe that, right? There is no way to really observe a gas coming from plants.
How do we see air?
Right? But until now, the puzzle looks like this. We need water, we need some injured air. It makes the plants grow, and it also gives off some good air.
Some good air.
Yeah.
Which we now know what it is. But this is very interesting because you are taking every one of us through a journey where we are actually getting into the shoes of these scientists and those investigators who did this, and now we are at some level of solving this puzzle through experiments.
Slowly we are there.
We are going there. But this is something interesting that you mentioned, because everybody was fascinated with gases at that point. Like right now, everybody is fascinated with microbiology and vaccines because of COVID and pandemic. So one of those people and scientists is what you're looking at here is Jan Ingenhousz.
Yeah.
So in similar time, he was very inspired and fascinated by Priestley's experiments. But he wanted to actually investigate what exactly goes in, what that gas is. Is gas being produced really? Because we can't see the gas.
Right.
Indirectly, Priestley is thinking there is something good being produced and which is being used by the mouse which is in the jar. Now, to tell you background of this particular scientist, he was not a botanist.
Okay.
He was a physician. He worked a lot on smallpox because at that time, you can imagine smallpox was really a problem in various countries, including India and England also. So being a Dutch scientist, then working in Britain, he was working a lot on smallpox disease and later vaccine also. But he is not famous for that. He is famous for actually understanding what gas is coming out, or is it gas that is coming out of the plants. He of course did a lot of experiment. He has published a book which is called as Experiment Upon Vegetables, where he shares his experience of doing 500 or more experiments on plants. But this is very interesting. What he did was he observed this particular plant giving out gas. Now what you are looking at actually, so if you want to think about how do we see gas, he actually had a brilliant idea. So he got all these plants, their leaves, different plant parts, put them in water and got it into the house. He used lots of glass jars, pickle jars, that we find at home to do these experiments. So what he could observe is some plants which were inside the house, like the Oxford Botanical House of Plants, he could not see bubbles coming out. Right. But accidentally he saw one plant which were near the window and it was in a sunlight area.
Okay.
And that plant was giving out lots of gas.
Interesting.
So
okay,
this
so
is
it was
interesting.
An accidental
completely accident.
discovery.
But then see, being a scientist and thinking logically helps always. He then thought gas is coming out. This is certainly, Priestley was clear that plants are giving out gases. But then what that gas is? Right? The easiest thing he did, he opened, initially, he took out a matchstick, he dosed it, he opened a jar can, that lid,
right,
and put the matchstick inside, and he could see immediately it's burning again.
Okay.
So then he thought it must be a gas which is useful for combustion.
So good air again.
Good air again, exactly. Okay. Now what happened after that is his observation, because he was noting down everything. He also observed that only those plants which are exposed to light are giving out gases, and those plants which are not cannot.
Interesting.
He went ahead, so of course he talked about gases, but he went ahead and then he proposed that it must be something in the sunlight.
Okay.
Which is useful for producing gas or perhaps growth of plants.
But then sunlight or the heat from the sun?
This is a very interesting question. Ingenhousz also thought about that. So at that time also we are thinking about what is sun, what it is giving, what is light, right?
Yeah.
Of course, most of the time sun is thought of our energy source. Right. It is thought of, giving that gives us heat and energy and so on. So to think about your question, we can also ask our audience, how will you find out that it is the light from the sun or the heat from the sun which is actually helpful in producing gas? What sort of experiment will
you do at home?
Right?
Yeah.
Just type in quickly and share what sort of experiment we'll do.
Until then, let's proceed to the next part, right? So now, what if we were Ingenhousz? What would we say? That plants kind of repair the damaged air or the bad air,
right.
And what else? Maybe until that time, later on, I think oxygen was really discovered. So they knew that good air means oxygen.
Perhaps it's oxygen.
Yeah. Okay.
But isn't this interesting? Because what is happening is plants are using something else and giving out oxygen as a byproduct.
Which is really important for all of us.
Yeah. So without plants, you can imagine none of us can survive.
Right.
Okay, so answers are coming in. I think one or two of you have got the correct answer. He did again a very simple experiment. He used 2 glass jars. One he kept in sunlight, and when he kept inside his house near a fireplace where there is lots of heat.
Right. And
And
the one
here
with
you
the
observe-
heat didn't give any
No.
oxygen.
So it's
Okay.
something in the sunlight which is actually giving plants the energy to produce this particular
Interesting.
gas.
That's
And
right.
this is said to be a landmark in our equation, which is now sort of
We
coming
need
towards
the
the end.
sun.
Exactly.
Okay.
So we need the sun, and not the heat, but the sunlight to actually do the process. Where now the equation becomes, or the puzzle becomes, there is water, there is air. Now we know it's a mixture of oxygen and carbon. With sunlight, plants are able to grow and produce good air, which is completely now known, sort of, which is oxygen.
Interesting.
Now going ahead, this is something that we have, studied, we have done in our labs, also sometimes in IISER. But do you think our students, our viewers can do something at home? Something similar that, Eisenhower did or Priestley did? Sorry. Something
Yes,
that
so
Ingenhousz did
we
or
can
Priestley
even
did?
try doing the photosynthesis experiment. So
How do you want them
to
to?
show that photosynthesis is happening and gas is being produced. So one way is the funnel experiment that we did, but here is something you can do quantitatively, as in you can measure the rate of photosynthesis by any plant.
Wow.
So what you're going to need for this particular experiment is a hole punch. You're going to need different plant leaves.
Okay.
You're going to need some baking soda. You're going to need a dish soap. So all of these things are pretty much available, and water, which is available at home.
So like Ingenhousz, our viewers can also do similar experiments.
Yes. So let's actually do this experiment. What we are going to do is we are going to punch holes in the
Okay.
leaf, and we are going to get these small discs.
Okay.
Right. So our aim is to get around 20 discs. So 10 discs for one experiment, 10 discs for another. Okay. Now that we have done that, what we want to do is, remember that plant already has some residual oxygen
Yes.
in the leaf.
Yes.
So first, let's get rid of that.
Okay.
So what we are going to do is we are going to take a syringe, fill it with water, and add our discs. So I'm going to put in 10 discs
Okay.
in this one. With the syringe, I'm going to suction some water in it, and then I'm going to create a suction pump such that all the air or oxygen that is in the disc is kind of removed. So as you can see, the discs were floating before. Now after this step, they are all down there. They're at the bottom of the syringe, and that is what we want to do, right? Okay, so that's for the discs. So now let us prepare our setup for photosynthesis. So what we are going to do is we are going to take two glasses, both having equal water. And what we are going to do in each glass is we are going to add a drop of the dish soap. Okay, let's just stir it. And then in one of the glasses, I'm going to add some baking soda. The other glass, let's call it the one without baking soda. The other
with
glass is
baking soda.
one with baking soda.
Okay.
And again, let's stir it. And now what we're going to do is we are going to empty the discs, 10 discs in each.
Okay.
Okay, so we want you to actually think, what is the baking soda and dish soap doing?
Exactly.
What are they doing? Why did we add them to the water? Right? What chemical equation will really signify what they're doing?
So another person within a person.
Yeah.
Okay.
So now let's put it in sunlight, and what we are going to observe is something like this, that as photosynthesis happens, so what do you think will happen now?
So like what we have seen so far, of course, plants which are in water, exposed to light, they should give out oxygen, basically small bubbles that we
Right.
can observe. But now since in one dish you have put baking soda, which is, you can put the chemical equation of baking soda in our chat, and in another you have not added. So what I think I can predict, and we can test it later, is small bubbles coming out of these small discs.
Right?
And perhaps if there is enough volume of gas which is being liberated, something will happen which you did exactly opposite of at the beginning. You took out those gases,
Right.
and they were settled. If there are again gases generating through the leaves,
So they will.
leaves should, or the disc should float up.
Great. So that's what you in fact observe. So let's look at those small tiny discs, rising up in the glass
Wow,
jar.
this is magic, isn't it?
Not only that this is a good experiment to try, but it is the reason why this is a good experiment is it can help us to determine the rate of photosynthesis now.
Right.
So the faster the disk goes up, that means more oxygen is being produced. That means the photosynthesis is happening at a very, quick rate or a
Yes.
faster rate. So in fact, that's what we want you to do at home.
Yes.
Count. You have added 10 discs. Now count the time it takes for 5 discs to go up. Okay, this time we'll call as ET50. Okay.
Okay.
And remember, ET50 is going to be inversely proportional to the rate of photosynthesis. I mean, if you want to make it simpler, inversely proportional meaning if the time taking for the discs to go up is really high, that means that photosynthesis rate is really low. But if the discs rise very quickly, that means photosynthesis is also very fast, right? So we are going to, really test out the rate of photosynthesis for various plants around us.
Exactly.
You can try different, ways or different ways to really manipulate this experiment. So what all experiments can you do?
So I can think about different sizes, different shapes. If that is going to make change, instead of a disc, I'll cut a star.
Yeah, will it
I
make
can
a
also
difference?
think about doing this with leaves which are exposed to sunlight, and maybe those leaves which are not directly exposed to sunlight.
Interesting.
And let's see if that makes any difference. So basically what we are asking them to do is become scientists at home and try out this experiment.
Do send us your results. You can also try like colored leaves and non-colored leaves. That's one
That
way
would
to
be
go
very
about.
interesting.
You can also try different light sources. So you can maybe, set up a lamp at home. A little fancier experimental setup is a lamp at home. Try to see if the closer the lamp gets to the disc, does it change the rate of photosynthesis. So many variations you can really do at home.
Exactly.
And probably try plants that are near your house, so
Yes.
and see if the rate of photosynthesis and see if the rate of photosynthesis for those plants is documented. See if you can contribute to creating knowledge.
So
in
this is what you mentioned, very interesting, Neeraja, because most of the times we just do the experiments which are already there in our journals and books, but this is something you are asking them to do beyond that.
Let's go beyond
And
that.
that is the way of doing science, right? Questioning and then going ahead. Great.
Yes.
Now I'll take you to the next level where we have clearly now said that sun is important. Of course, there is gas and there is soil, water, and they are producing something. Now in the equation, the other part of the equation is what comes out of this whole process, right?
Right.
Now, of course, after that time, around early 1800s, there were many advances that happened in understanding plants and animals and everything around us. Even microorganisms which we were not able to see before that. So how we started doing that is invention of microscope.
Interesting.
So I'm going to tell you a story of Julius von Sachs, right, who tried to answer this question, why sunlight is so important. Sunlight is important, we know, but why it is important, how it is helping the other side of the equation come to the existence. Now I should mention this. Sachs should also is given a lot of credit in our understanding of how plant cells, plant tissues works. So he worked extensively throughout his life on plant physiology. So our students who are doing in 8th, 9th and 10th standard, they know about plant tissues. They know about transportation happens in plants, how food is stored, how food is transported, everything. So he is the person who started all this investigation. Of that, one investigation I am going to share with you is till this time we knew that sunlight produces gases from leaves and gas is the oxygen. But then what is getting converted into this process, that is getting stored in plants, right? During that time, along with Sachs, there were other scientists who were working. So for example, around 1883, Arthur Meyer, so mind you, this is after let's say 100 years from what we have seen so far, actually used microscope to see what is there inside plants. So he could actually see these small green granules which are very tiny. By the way, what you are looking at is actual drawing that is made by Arthur Meyer from Germany. So he called these bodies as chlorophyll corneae.
Okay.
which we now know as chloroplast granules. But along with him, before him, around 1790s, Comparati and few other observers also noted that these green granules are present in all plants, almost all leaves. Right?
Interesting.
It will be interesting to see whether yellow, red, or other
Different colored
plants also have this, but you can find that out. Around 1862, Julius Van Sachs demonstrated light-dependent starch formation. What does it mean? Basically, he used microscopy and lot of other patient observations to actually look into what is happening inside a leaf.
Wow.
The video that you are looking at, you can actually see the discs that are floating inside plant cells are actually chloroplast.
Wow.
And then he used different chemical tests, also microscopic observations, to come to a conclusion that this particular storage of food, which is there inside, is actually starch. But since now you and our listeners are also actually noting this, we are not only talking about biology. We are talking about chemistry, we are talking also about physics, right?
Yes.
So what sort of experiment you can think of, Neeraja, to come to this level that along with water, air, sunlight, and something which is green in plants, is helping plant grow and also produce good air?
Right. So, I mean, the way I would think about is that what are the plants really producing?
Exactly.
And I would probably do a lot of chemical tests that are available at that time to really figure out what is the constituent of the plant.
Exactly.
Right. So, in fact, we can even try this at home. So let's see if we can do.
So again, another
what
experiment
Van Sachs did.
Exactly. Nice. Nice. Okay.
So we are going to do something that he did, which was to take a leaf, and remember that chloroplasts are really important for the sugar to form or the starch to form in leaves. Let's try to block out a part of the leaf.
Okay.
Okay, so now that part is not getting any sunlight, and that part will not do photosynthesis, but the rest of the leaf will.
Now this is only possible because scientists before him actually solved the other part of the puzzle, right? So this is actually, you are asking them to be Sherlock Holmes, is it? I think so. Okay.
So what we are going to do is we are going to put this leaf in alcohol. Why alcohol? Because it's going to dissolve all the green part.
Okay.
And basically chloroplast will not interfere with our reading.
Okay.
So what they are going to do is they are going to heat it in water.
Okay.
Dissolve, basically create a water bath, add a test tube full of alcohol and the leaf, let it boil such that all the chloroplast comes out. And then what you have remaining pretty much is the starch.
Yeah.
In the leaf.
Just a word of caution, don't do this alone. You have to do this with your parents or teachers only.
Yep. And in fact, now that we have what we think is starch, let's add some iodine. So that's what Van Sachs did, is he added some iodine and saw that the part that was exposed to sunlight is turning really dark blue-black. And in fact, that is the starch iodide complex that is forming.
Right.
In fact, this is the same reaction that you learn under addition reactions in your chemistry textbooks, right? Which is when starch and iodine combine, they are really adding together to form a complex that looks blue-black in color. And what will happen to the part that was covered with black paper then?
If we go ahead with the understanding of how photosynthesis works, no sunlight, no production of starch.
Great. So that's what we observe, is that still remains the original color. So that's how we figured out that there is starch that is being produced.
That's brilliant. So you are using chemistry to solve biological problems, is it?
In fact, that's how most biological problems were solved, using physics and chemistry around us. So in fact, we want you to try this at home. There are various plants around us that have multiple colors. So they have, for example, this plant right here, it has green color, it has yellow color. What happens when you do this same test with such leaves?
Because we are talking about chloroplast, which is green.
Green. So we want you to try it and tell us what happens.
So the question we are asking is, how do color plant perform photosynthesis? Because we don't see them as green.
Right. So let's try to answer it in the chat box then.
Exactly.
So there are various things that we can think about. So let's take the example of red amaranth and green amaranth. लाल माठ, हिरवा माठ, जे मराठी आमचे viewers बघतायत त्यांच्यासाठी. So the red and the green, two different colors. You think there is chlorophyll in the red amaranth?
If we go by our equation, it has to. But then you are taking me back to the initial experiment, obviously, and before that, where we are talking about, is it really air or water or soil which is asking or which are helping plants to grow?
Right.
Perhaps in red plants, it's soil that is helping them to grow.
Probably, or there are other parts of the plants like a stem that is green.
Which is green, yes.
Right. So in this case, for example,
Yeah, you can see green here.
right. So for example, this leaf performs photosynthesis, but not this one, which is red in color, right? So how do we know? So what do you think is happening? There are 4 options on the screen. One is that there is a lot of red pigment, which basically masks out the green pigment.
So there is still chlorophyll somewhere, but
Yes.
it's actually red at the top. Okay.
The other option is that the red pigment is really small amount. The green pigment is really huge, and that is what still does the photosynthesis. The third option is that there is no photosynthesis happening in the leaf, but probably in some other areas of the plant. So for example, in tomatoes, the green tomato, for example, can do photosynthesis, right? Perhaps. And the other thing is that there is enough chlorophyll, there is enough red color also, and it's just getting masked. So let's see what our viewers think, which one they think is true.
Okay.
Okay, so we see a lot of distribution of answers really.
But Neeraja, are you going to give them an answer or ask them to do an experiment?
I am actually going to ask you to find out. So if
Okay.
you were to find out, what would happen? How would you do this? So I'm going to give a clue. Maybe you can try this at home. Is one thing we are going to do is we are going to do chromatography. So all of you have done this. Chromatography is mainly used to see the contents in a mixture. What
Okay.
all things are there in a mixture. So let's see this chromatography of black pen, for example, sketch pen. As the water rises on the filter paper, what we see is that it's made up of 3 different colors.
Okay. When you say chromatography, I thought it is something very complex.
Oh,
But actually
it's
not.
a fun thing
Yeah.
that we all have done as kids, right?
Yeah.
So the same thing we want you to do with leaves now. So I'm going to show you the chromatography of a spinach leaf here.
Oh, nice.
So what we are going to do is same thing, filter paper. We are going to take a coin and scratch spinach on the edge of the filter paper and let it rise up in various solvents. So you can try it in water, you can try it in alcohol. So rubbing alcohol, isopropanol that you get in the chemist shop.
So the same alcohol that they have used for our starch experiment, they
They
can
can
use the
use
same.
here, right? And in fact, you can do mixture of water and alcohol, various mixtures. See which one gives you the best chromatography,
Okay.
chromatograph, right?
Okay.
And what we should see ideally is that the pigments in spinach should really separate. So carotene going up, chlorophyll A, chlorophyll B. There are various pigments.
Yes, which we study in textbook.
Yes,
Indeed.
that are there in the textbook. So we want you to
Okay.
try this with spinach, but we want you to go further and we want you to try this with something that is red leaf,
Also.
right? So we want you to tell us what all pigments are there in the red
Yes.
leaf.
So along with chemistry, let me also put in some points from biology.
Sure.
We can observe this also. If we have
Okay.
microscopes, so in school or fold scope and
Interesting.
microscope, we have done an episode, Neeraja, where we have shown how household things can be used to generate a very nice microscope. If you have a mobile phone, then nothing like it. So I'll talk about that. What they can do is, using microscope, along with the experiment that you have just asked them to do, is actually look at the plants and see how does this gas exchange, which is at the core of all this, happens. So I'm sure you must have heard of stomata. If not, you can hear it maybe next year when you go to the higher standards. So you can actually see how stomata works. You can
Okay.
actually see stomata which are open, which are closed on the underside of the plant, of the leaves, right? To do that, again, we require something that is completely available at home. What we require, of course, few leaves. We require a nail polish.
Okay.
Right?
Clear one.
A
I
clear one.
see.
Ideally, very light, very transparent one, not the dark
Okay.
one. At the same time, we require a glass slide. If you don't have a glass slide, you can use a very clean sort of new plastic scale, which is transparent, and a sellotape. Nothing
Okay.
else.
Okay.
What you need to do is you need to put some small amount of nail polish on the backside of the leaf.
Okay.
Then take a piece of sellotape, put it on top once it is dried, and then in one quick action, take out like you do in band-aids.
Okay.
So take out,
Ripping off the band.
ripping off the bandage. Take out the sellotape, and there you have it. You just have to put it back on the glass slide or the scale that you have. And then you can use Foldscope, you can use Microscope if you have at school or at home. You can also use different types of microscope that we are showing in this video. You can see the link below to actually see how these openings look. And you can at home, you can see those openings.
So these are the openings where the gases that we were
Exactly.
talking about really,
Exactly.
pass through, right? Or
Exactly.
get into
Exactly.
the leaves. So
Exactly.
is that how CO2 gets into the leaves?
We can ask them to find out.
Okay.
So of course you can design experiment. You can also see few videos how to do this. Interesting thing is, so this is something we want you to try out at home, because this is actually putting all pieces of the puzzle together, right? So I know stomata has directly nothing to do with photosynthesis, but without these gas exchange, you won't be able to carry out the whole process.
Right, and I think stomata is very important for a plant to have open stomata. let's say,
Exactly. Now this is something you can try, but you can ask more questions. So see, now we are coming from answering a puzzle to asking more questions, and this is how the process goes. You can think about different types of leaves which are from a small plant or old adult plant, leaves which are very young, which are not very young, and you can just compare what different types of stomata, whether they are closed, whether they are open, are they closed only in day or are they closed only in night. You can answer all these questions by doing the same method, but actually taking different samples.
Yeah, and in fact, I was thinking the plants underwater. Will
Why
they have
not?
it closed or open?
Or will they have stomata at all?
That's really interesting. So let's try this
A
at
lot
home.
of homework for you.
Yeah.
But mind you, we want you to send these photos, picture, your experience to us. And then we'll share a link on which you can share all this. And we want to use some of your experiments, some of your results to be shown in our next episode. We'll show you that.
Yes. So in fact, now we have come very close to at least this particular equation. Is that
Yes.
we need carbon dioxide, we need water, we need light, we need the plant chloroplast
Yes.
to give us the sugar, the starch, and oxygen. Right. So that's how the whole each piece of the puzzle was
Is
really
there.
weaved. So remember, we started from 1600. Now around 1893 was the first time the term photosynthesis was really coined by Charles Barnes, and he in fact called it photosyntax.
Syntax.
Later on moved it to photosynthesis. And see how we've traveled for 200 years to come here, but the 300 years, but the journey doesn't end here, because
Yes.
it goes on. Because remember, if we add carbon dioxide and water together in a flask outside, add light to it,
Yes.
it doesn't really create sugar and oxygen.
Yeah.
So what is the plant doing inside?
What is exactly happening?
What's
So,
inside the leaf that
Leaf
is really
that
doing
is
it?
happening. And I think by this time we have, actually very sophisticated technology. Science has progressed a lot. By this time we have actually gone down to understanding an atom also. At the
True.
same time, gone out in the outer space and looked at many satellites and many planets and so on. But I think the next part that we are going to share, which is in the previous century, actually led to winning a Nobel Prize.
Yes, so this is the work by Kelvin and Benson, and Blackman for that matter, which talks about how plants really convert. What happens inside the leaf that converts CO2 to oxygen.
Yeah.
And to think about it, they had to wait a long time. until really radioactivity was really discovered.
Okay.
So until we got an idea about radioactivity and how C14 or carbon 14 really, the isotope of carbon really degrades and how we can track that degradation.
Okay.
That's how that's the idea that Benson used. Now to think about it, imagine that it's a black box, right? What is happening inside the leaf is a black box.
We can't see inside.
You can't see inside. What is going in? Carbon dioxide. What is coming out? Oxygen. So what is happening inside?
Yeah.
You really have no idea.
So where the oxygen is going and where the carbon is going.
Right. So now we are going to add a tracker with the
Okay.
C14 or the radioactive carbon.
Okay.
And because of that, we are able to track where the C14 is going inside the box.
So this is something very similar to when we share our location on Google Maps, right?
Right.
We can actually track where we are going.
Right. So what he did is this lollipop experiment.
Lollipop.
So the reason why it is called lollipop is because look at the shape,
The shape of the glass.
glass.
Okay.
It looks like a giant lollipop, right?
Okay.
And this is the famous lollipop experiment. What he did was he filled this glass with blue-green algae.
Okay.
And blue-green algae, just like plants, does a lot of photosynthesis. Now perhaps faster, yes. So now to this blue-green algae, what he is going to do is he is going to feed the carbon 14 CO2.
Okay.
Okay.
Which is radioactive.
Which is radioactive, and we can track it.
Okay.
And what algae is going to do is it's going to start doing photosynthesis. But what we are going to do is we are going to say, okay, you started the process, stop.
Okay.
We want to see how you have progressed.
So like stepwise.
Yeah. So we are going to kind of clicking pictures every now and then to see where the carbon 14 is going.
Okay.
So what he did was he fed the carbon dioxide, and then he every 5 seconds he is going to kill the algae really to figure out where algae has reached.
Okay.
You know, so let's say that you're going from Pune to Bombay. You're kind of stopping every second and figuring out, so just like Google Maps, where you are. And that's what he did. So and the method is really simple. It's
not
something
actually clicking photos, right?
not actually clicking photos.
Yeah.
But the method is the same as doing chromatography. So in fact, he did chromatography too, and he figured out that at 5 seconds, these are the molecules formed inside the algae. And then at 10 seconds, these are the molecules formed. And then at 30 seconds, these are the molecules formed. So he's trying to really capture an image, and then later on trying to weave these images together to figure out like a flow, like what is happening, where is this molecule going, is it forming this or this. So it's not an easy experiment to do. It took them around 10 years to figure this out, just with simple chromatography, nothing fancy, but chromatography leading to the answer of what all chemicals are formed as
the carbon dioxide coming in and
oxygen going out. So that's really the beauty of this particular experiment. In fact, now we know the Kelvin cycle. So to think about it, we have one carbon that is coming from carbon dioxide. And imagine this, that we are getting 6 carbons out in the form of the glucose that we are forming, the sugar. So what is happening here? Let's think of it like plant being a factory. Right? So it's the leaf, each chloroplast is kind of a factory doing some setup together. So it's going to grab a carbon dioxide from the air, and what it has is this 5 carbon compound, rubeose bisphosphate, and in fact rubisco is kind of the enzyme. So the person handling the factory, let's say the enzyme, is going to get these two together and form a 6 carbon molecule. And you would think that, okay, we have 6 carbon molecule, glucose also requires 6 carbons, the job is done. Not quite, because we have to convert this 6 carbon molecule to glucose, right? And how it does is it first breaks the 6 carbon molecule
into 3
and 3. So you have now your phosphoglycerates. And these phosphoglycerates are again reacted with NADPH and ATP, which is the energy source really, to add a hydrogen to this, forming glyceraldehyde 3-phosphate. Okay, so now what we have is 2 molecules of glyceraldehyde 3-phosphate. Together, how many carbons do we have? 6. Now I can combine these to form glucose.
Yeah.
Right? Is that correct?
But yeah,
I think something is going
wrong
wrong
here.
here, right? So what could be the wrong thing here? Remember that the plant is doing this again and again, completely, constantly, like in a cyclic manner. So every time if the plant has to, consume 5 carbons from this rubiose bisphosphate, there is a deficit of 5 carbons now.
Yes.
So how do I really
get the new one for the next reaction.
the next reaction? So I cannot consume something just like that. I have to also reproduce it so that it is available for the next cycle. And in fact, that is what the tree does, is that it doesn't just run one factory. It's going to run 6 factories. So altogether, instead of
making parts for the whole thing.
So instead of having just 2 molecules of the glyceraldehyde 3-phosphate, now you have 12 molecules, total 36 carbons, out of which it is going to take out 6 carbons.
Yeah.
So you have 30 more remaining. And those 30 it's what happens is that once in the step 3 we form the carbohydrate, those 30 are going to go back and reform those 6 RUBPs, the rubiose biphosphate, that you need for the cycle to keep running. And this was not an easy task to figure out. It took us a long time. In fact, I think we still are figuring things out about this particular process.
Exactly. And I think who is going to find those answers is our next generation scientist.
Yeah, so this is the puzzle, like what
Neeraja,
happens.
thank you so much, because the way now you have explained Calvin's cycle and where we start and where we end, I think I need not again mug up Calvin's cycle. It's actually more interesting.
Right. Now think about and as you advance in sciences, maybe you can think about the actual chemical structures.
Exactly.
What reactions are happening? How are they happening? How can we make them faster, for example?
Exactly.
Right? So that we can produce more oxygen, which is the demand of this time.
So plants are brilliant in every sense, it seems.
Yeah,
right?
they're
And
amazing factories.
Exactly, amazing factories. And just to give you an idea, if you want to compare this, look at this, right? This is the same mango tree we started with. So average rate of energy captured by photosynthesis, if we talk in terms of now energy in watts, right? It is approximately 130 terawatts, which is equal to 8 times the power we consume across the globe by whole human civilization.
Wow.
So you see how much power they have, and this is actually coming from sunlight. So we are actually using, if you are using plants, the energy which is coming from sunlight. If you go ahead and look at this, photosynthetic organisms, not this plant. So since you mentioned algae in the last experiment, convert around 100 to 115 billion tons of carbon into biomass per year.
We really need plants.
We absolutely need plants, and in good condition. And these are ready-made factories. You don't have to put any other factory, right? Just to show you, give you a comparison between the distribution of all global biomass. Right? This is from World in our data. So total global mass is around 546 tons. This is a bit older value. Of that, 82% is only plants. Next higher is bacteria around the globe, which we don't see.
Right.
Then fungi, then archaea, which is sort of a specialized type of prokaryote. And then if you can see here, the small square here, which is now zoomed out, comes animal biomass. Right? And within that, this is us.
Wow.
Right? So perhaps you have to look at, all the life around us in a very different way. But that leads us to solving the puzzle, which took almost 400 years, and this continues, of doing science.
Right. So in fact, we are still solving this puzzle, trying to solve, right?
So see these questions now, which are still unanswered, which is again related to the process that we have started. So what are proteins? What are enzymes? What are their roles in the whole process? Think about evolution of different types of photosynthesis, because different organisms, even different plants, use different set of machinery to carry out photosynthesis.
Wow.
Think about the energy dynamics. Since you now think in terms of energy, think about energy dynamics. What sort of energy dynamics is happening within this, right? How does it work in different species? How does it work when plant is small? How does it work when plant is large? Different types of photosynthesis also we know. So what sort of reactions are happening? And at the end, designing artificial photosynthetic system, if we can do that.
True, because I think, what do photosynthesis result in? It results in oxygen and really cleaning out carbon dioxide from the air.
And this is going to help to solve new age challenges. So the pollution that is happening, which is growing, the food crisis, right? India, few decades back, started the Green Revolution, right? So that has helped to tackle these food crisis which was there immediately after our independence. Increasing atmospheric carbon dioxide concentration, energy crisis, and so on. And many scientists in India and across the globe who are working on this. We should mention Sir Jagdish Chandra Bose, who contributed a lot in the 20th century in understanding photosynthesis and going ahead. So then, could photosynthesis solve these problems? You have to think about this. We'll go ahead and just pose few newer advances that has happened in photosynthesis. So boosting photosynthesis in crops is being now studied in places like IISER and other research institutes across the country. So artificial photosynthesis is also being tried on how we can generate the same amount of energy or food through artificial photosynthesis, and also basically trying to try to solve the challenges and problems of climate change which is happening through the brilliant species, the brilliant organisms that we have been studying in last 1 hour, which are plants. But this doesn't end here, right? We also want to focus on the core process of science investigation that we have been highlighting.
In today's episode, I think we went all over the process, right? We started with the question, what makes plants grow? Then we tried to do some experiments. Then we tried to figure out so piece by piece, how do we really put the scientific information together? So what do you think happens? What is the core process of scientific investigation?
So for that, we are going to ask you to draw a flow chart and share with us what steps do you think actually take place in the process of scientific investigation. You can use different problems that we are posing here.
Right.
Maybe a problem of understanding space and what goes on there. A problem of finding relationship between species on Earth. Right?
Wow.
Solving environmental problems, thinking about green chemistry, identifying patterns in nature. Why can't you use mathematics in doing that, right? Or making our lives healthier, tackling COVID-19, or developing vaccines. So these are the problems that we are still tackling and we are trying to find out answers to. But perhaps when we think about the process of scientific investigation, it is perhaps the same, right?
So what do you think? So all these could be solved, let's say, with scientific investigation. But let's try to figure out what goes in the process. So in fact, looking at today's episode, what do you think is the process of science? Which steps are really important to start doing as you start solving a problem? What all things that you do as you solve the problem? Which questions would you ask? Which puzzles would you like to see? So please fill the form, that is the process of science that we are going to share in the video description. And we would love to see your answers. In fact, we would love to talk to you more about your answers. If we find something really interesting, we would call you here in Science Activity Center and would like to help you investigate that part. So investigate it with us. So all the school students, all the college students listening to us, please write to us. In fact, teachers also, if you want to investigate something, write to us, fill the form of process of science.
Nice. So we leave you with this interesting quote. We don't know who has quoted this, but see this. So the crux of science, as we know it, lies in the details of the process, not just a snapshot that are present in our textbook. What goes behind this is equally rather more important. Right? It is important to study process of discovery, not just memorizing the discovery itself. And that is what we are trying to do here, right? Especially on the special occasion of Children's Day.
Right. I mean, that's the idea, right? Don't just mug up the equation of photosynthesis, but try to understand how we came to know the process of photosynthesis. So if we can adopt this idea for all the things that we learn in the whichever subject that you really are learning in school, I think we'll be good learners.
Great. So please reply, please send your responses. The links are given below. The other links of our videos, the video that you saw throughout, and the microscopy video is also there, right?
Yes, send us the results of those experiments as well.
Absolutely.
We look forward to your answers. So on the occasion of Children's Day, we want to wish you all a very happy Children's Day. Keep exploring.
Keep being a scientist.
Yeah, keep the child in you alive. Thank you.
Thank you. Bye.
Bye.